Hi everyone. We're gonna be talking about control of eukaryotic genes this time. So we talked about control of prokaryotic genes last lecture. Now it's gonna be eukaryotic genes. Um, Basically, the big questions of both prokaryotes and eukaryotes are simply why or how are genes turned on and off, and how do cells with the same genes differentiate to perform completely different specialized functions. Because that's the case of a multicellular eukaryote. We have a whole bunch of cells in our bodies, and all of those cells contain the same genome, but not all of those cells produce or express the same genes or produce the same enzymes. So, how can they specialize their functions? Our liver cells are not going to do the same things or produce the same enzymes as our neurons. We're probably already remembering a little bit of your last Khan Academy reading with these questions of how eukaryotic cells control their genes. Um, Prokaryotes basically evolved gene regulation more than anything to quickly adapt and respond to their environments, right? They need to be able to adapt to the signals produced by their environments quickly so that they can survive. Eukaryotes, we of course also need to adapt to our environments and respond to our environments quickly, but we also need to regulate the complexity that is all of our cells in our body, and how we can regulate that with growth and development, and we can regulate specialization, um, just turning some genes on in some cells and turning off other genes that are not needed in those cells, and vice versa for other types of cells in different organs or different places in your body. Um, how can we control gene regulation in eukaryotes? There's all of the central dogma. Remember, central dogma states that DNA first is transcribed into mRNA, and that mRNA is then translated into protein, and that's how we can get all of these proteins or these enzymes from that basic DNA code. Okay, thinking about that step by step of DNA unwinding, being able to transcribe it into a messenger RNA molecule, and then being able to use that messenger RNA molecule to translate it into a protein. All of those steps can be regulated. So all of the points of control for gene regulation are those steps. For example, the packing or unpacking of DNA, we're going to talk about that in a second. The actual transcription of that DNA into the mRNA. We also know that the first primary transcript is not actually the mature messenger RNA that is used to produce the proteins. There is processing, this processing and splicing of mRNA. So you can regulate that section, that step. You can also regulate mRNA transport. A lot of the times the mRNA from the nucleus needs to go to not just the cytosol, but different parts of the cytosol. So you can regulate that transport. Then you can regulate the translation of that mRNA into protein. You can also regulate protein processing. We're going to talk about that hopefully. And you can regulate protein degradation. You can make a protein degrade before it fulfills any function. So in that way, you're regulating genes and the expression of those genes. So first of all, DNA unpacking. We know that DNA can be packed super tightly into this chromatid form. Remember that in DNA replication, DNA replication and mitosis, we were talking about how you can see when the chromosomes actually reach mitosis, reach interphase, reach prophase, not interphase, reach mitosis, which is prophase. They coil up into these dense state. And if you coil up into this dense state, it is very unlikely, very difficult for the DNA that is in that chromosome to unwind enough for a RNA polymerase to be able to go and do its job and replicate that DNA transcribe that DNA into an mRNA. So you need to have that DNA be unwind, or you need to have that DNA be loose so that an RNA polymerase can actually get there. So coiling or uncoiling the DNA is one form of gene regulation. When you're talking about how a DNA is wrapped, you can talk about nucleosomes. So nucleosomes are the first level of DNA packing, and they involve histone proteins. So it's this DNA, it's this histone protein, and then the DNA is kind of wrapped around this histone protein. So the key here is that these nucleosomes can be tightly packed one after the other, or there can be pretty much stretched out. Okay, so depending on if the nucleosomes are tightly packed one next to each other, that DNA is super coiled up and super condensed. If the nucleosomes are kind of elongated in a string-like formation, then that DNA is very uncoiled and very easy to transcribe. So when you're talking about this easy to transcribe DNA, you're talking about euchromatin. Okay, if you see right over here, euchromatin is going to be loosely packed DNA where the RNA polymerase can easily reach the DNA and start transcription. If the DNA, because these nucleosomes are so tightly packed, is tightly packed, that is called heterochromatin, and heterochromatin is usually where transcription is not seen, because this tightly condensation of DNA makes it impossible for the RNA polymerase to reach the DNA and to transcribe those genes. Another thing that can be used in this state of uncoiling or coiling the DNA to regulate gene expression is something that's called methylation. So it's the attachment of a methyl group, usually to cytosines. It can be attached to other nitrogenous bases, but it's usually to cytosines. And when you methylate a particular segment, you make it so that it is very unlikely to be transcribed, extremely unlikely to be transcribed. So what you do is pretty much you inactivate these genes, the genes that are in the segment that has been methylated. And most of the time, this is nearly permanent. So we've seen this in cats. Remember that we were talking about female cats having two X chromosomes, right? Because they're female. Sometimes one of those X chromosomes can be methylated, and this will be at random. So depending on what cell has methylated what X chromosomes, all the cells that come from that cell, all of the cells that become the progeny of that one original cell, will have that X chromosome methylated or inactivated, and that inactivated X chromosome is called a Barr body. The cells, for example, that originated, the cells from the back, let's say the back of the cat, that originated from an X chromosome that was methylated, let's say that that cat had two alleles, one for orange color, one for white color in both X chromosomes. If the X chromosome for the orange color gets methylated, then that segment of the cat's back is going to be white. But then the cat's face could have methylated the other chromosome. The white X chromosome becomes the Barr body, so the cat exhibits the phenotype of orange color in his face. So all of these patches, in her face actually, because it's a female cat, all of these patches are based on this methylation pattern. We can also see in opposition to methylation, there's acetylation. So you can acetylate histone proteins. So basically what happens is that this is a nucleosome. Okay, so right over here, this is a histone protein, and this is the DNA that is being that is wrapped around this nucleosome that is made up of these histone proteins. So the histone protein is going to have a tail, and this tail is usually positive. It has a positive charge, and what it does is that it makes this nucleosome, attracted to another nucleosome. So it condenses one nucleosome with the other, all attracted to each one of the adjacent nucleosomes, and it makes this DNA super coiled up. When you acetylate this histone tail, you make this charge neutral. You neutralize this charge. So what you're pretty much doing is you are taking away the attraction of this nucleosome to its adjacent nucleosome. So you're uncoiling the DNA, and when you uncoil the DNA, you make it easier for the RNA polymerase to reach the DNA to transcribe. So acetylation, when you have an acetylated chromatin, that means that this acetylated chromatin is going to have its genes turned on. If you have a condensed chromatin or an unacetylated chromatin, that means that the genes are turned off. Okay, so just based on the attachment of acetyl groups to these histone tails. Okay, transcription, you can also, this was all prior to transcription. Now you can actually control transcription or regulate transcription through the promoter and the enhancer sites. So this is very similar to what we've learned with operators. Okay, the promoter is also where the RNA polymerase is going to bind, and it can only bind with something that you guys already talked about, which are called transcription factors. Okay, so when the transcription factors bind, they allow the RNA polymerase to bind, and transcription may begin. Again, if the promoter is in a region of DNA that is super coiled up, super condensed, then the RNA polymerase is not going to be able to bind. If the RNA polymerase cannot bind, then transcription cannot occur. Okay, the enhancer sites, the name enhancer is really important because it is not in the promoter. It's actually something that's called the distant control element. It's right over here. Okay, so the promoter is very close to all of the coding regions of the gene that you're trying to code for, but the enhancer is actually pretty farther away. It can be very far away or just a little bit farther away, but that's why it's called distant or distal. In your book, I think it says distal. And what you can do is you can bind activators or you can bind repressors to these enhancer sites, this distal control element. And when you bind it, what basically happens is that this segment of DNA in between the enhancer and in between the promoter kind of bend. And what that does is that the activator gets very close to the transcription factors of the promoter. And what it pretty much does is that it activates or it promotes the attachment of an RNA polymerase. So the RNA polymerase is going to want to attach if it already has these transcription factors, but it's going to want to attach much more if it's got these activators. And many times these activators, what they also can do is that they can have different molecules that allow the segment of DNA near the promoter to be uncoiled, to become uncoiled. So if it becomes uncoiled, then it means that it no longer inhibits the binding of RNA polymerase, and the RNA polymerase can bind and can transcribe that gene. This is just what we've been talking about, this bending of this DNA segment right in between the activator and the promoter to be able to enhance the RNA polymerase and be able to initiate transcription. You can also regulate gene transcription or the regulation of genes, after transcription. So once you've already created an mRNA, remember that the first thing that you create is a primary transcript. Okay, so it's an mRNA, it is, but it's immature. It's the pre-mRNA, and you need to do a whole bunch of things to it. One of the things that you do is mRNA splicing. If you remember that you take on, you keep the exons and you take off, you splice off or eliminate the introns. But that completely depends on what you think are exons and introns. Okay, so sometimes you can be keeping just some of the exons, like in this case, this exon, this green exon color-coded as green here, is kept in this particular mRNA, while this exon that's color-coded as brown is kept in this other mRNA. They're the same transcript, they were coded for by the same gene, but based on what is being eliminated and what is being kept, you can create two different mRNAs. If you create two different mRNAs, you can also create two different proteins. So that post-transcriptional control is also super important. You can regulate also the processing of an mRNA. Remember that you need to add a GTP cap at the 5' end and a poly A tail at the 3' end. This poly A tail and this GTP cap allows this mRNA to exit, first of all, exit the nucleus and go into the cytosol. But as you guys recall, in the cytosol, there's a lot of nucleases that, or exonucleases that are trying to break down this mRNA. And if they are able to break down this mRNA no longer functions, right? So the length of this tail, for example, how long this tail is going to dictate how long this mRNA lives. So if you want an mRNA that only serves a very particular purpose, a very particular enzyme that you need to regulate pretty fast, and you don't need to have that much of, maybe you want to have a poly A tail that is super short. And if you want the mRNA to last for a very long time, maybe you need to have a poly A tail that is super long. So that's also a very a different way that you regulate the gene transcription and translation. There's something that you probably haven't heard of, and this is called the RNA interference. So you can also regulate the mRNAs that are being translated into protein by simply targeting different mRNAs that you do not want to be translated into protein and destroying them. So you can do this with something that's called the small interfering RNAs, and they're these very short segments of RNA that can be 21 to 28 bases long. And what they do is that they bind to this mRNA that you do not want to be translated into protein, and they pretty much tag it for destruction. So it triggers the degradation of that mRNA and causes, although the gene has already been transcribed into an mRNA, it causes the silence of this gene because the mRNA gets triggered for death or triggered for destruction and never actually gets to produce a protein. So it never actually gets to express the gene. So the gene is silenced. And we believe that this was actually a mechanism, that this evolved as a mechanism that prokaryotic cells, we can see this in prokaryotic cells, use this in order to try to target different genetic information of viruses that have their genetic information in RNA, so that the minute that the virus inserts their RNA genetic information into that cell, the cell can produce these small interfering RNAs and target that virus's RNA for destruction. And they do not need to be lysed, and they do not need to be infected, and they survive this virus. So that's one of the ideas as to how this evolved. Okay, you can also control translation. Okay, so you can block translation. So remember that translation occurs when the ribosome binds to this messenger RNA and it moves downstream from the messenger RNA and translates each codon into a particular amino acid. So that's translation. Just like you can activate or you can inhibit the RNA polymerase when it is transcribing into an mRNA, you can also activate or inhibit the ribosome when it's translating into a protein. So there's different sites where you can prevent the attachment of ribosomal subunits, and you can block this translation of mRNA to protein, or you can activate this translation of mRNA to protein as well. I think last thing that we're going to see is how you can regulate gene expression through protein processing and protein degradation. You can regulate, remember that proteins are extremely complicated, and there's a lot of proteins that are not just amino acids, and there's different things that need to be added into these proteins. So there's moments in time where you can regulate the transport of proteins, because remember that if you're in the endoplasmic reticulum, you can go into the Golgi apparatus to add different sugar groups or add lipids or things like that into the protein. So you can target the transport of this premature protein and not allow it to get to the other organelles where it can be processed and it can actually become the functional protein. So in that way, you're eliminating the functionality of the protein. You can also, once the protein is functional, you can also target its degradation. You can kill it before there's enough protein to actually make a difference in the cell. So how can you do this? You can actually mark it for destruction. So there is a very small protein molecule called ubiquitin. And what you do is that this ubiquitin can attach to the protein that has to be degraded. And once the ubiquitin attaches to this protein that you want to degrade, it takes it into something that's called the proteasome. It's also a complex of proteins. And within this site, it degrades that protein. So it creates just these very small segments, very small peptides. Okay, and once you've degraded that protein, it can no longer function. So it's kind of like if you silenced the gene that coded for that protein. So this is just a very small summary of what we've learned so far. 1 and 2, stages 1 and 2 have to do with transcription because they have to do with how you pack or unpack the DNA to allow RNA polymerase to be able to bind and initiate transcription, or you inhibit RNA polymerase from binding. And the transcription factors from the enhancer sites and the promoter sites, and how different transcription factors attaching to those sites will inhibit or will activate the transcription of that DNA, of that gene into an mRNA. You can also have post-transcriptional regulation, 3 and 4. If you are talking about alternative splicing, you're talking about right over here. And if you're talking about changing the 5-cap and poly-A tail, or the breaking down of siRNA, small interference RNA, you are looking at right here. If you want to talk about translation, how that works out in gene regulation, this is stages 4 and 5. You can actually block the start of translation through mRNA processing, or the initiation of translation right over here by having things block or activate the ribosome. Okay, and post-translation means that you are targeting that protein. So you can target the protein from not being able to be processed, so you do not actually create a mature functional protein, or you can target that protein after it's been mature and functional. You can target it so that it can be degraded with ubiquitin and proteasome. So hopefully that makes sense. Any questions, please, as you guys know, put it in our discussion board, and I will make sure that I answer them to the best of my abilities. And hopefully that was helpful. Thank you.